It's a beautiful day to learn about science. Today, we're diving into the fascinating world of recombinant DNA. You might have already learned about DNA, the blueprint of life. But what happens when we combine DNA from different sources? That's where recombinant DNA technology comes in. Today, we will correlate how this technology explains the mechanisms of sex linkage and genetic recombination, which also gives us a better understanding how traits, especially those linked to sex, are inherited. So, without further ado, let's explore anew and get right into it. Let's begin with recombinant DNA technology. Basically, this technology produces DNA that has been artificially combined using in vitro manipulation from 2 or more different sources. How come? Think of it like mixing and matching Lego bricks. Scientists can take a piece of DNA from one organism and combine it with DNA from another, even from different species, creating novel genetic instructs. A common tool used in this process is the plasmid, which is a small circular piece of DNA found in bacteria. We can cut the plasmid open using special enzymes such OSE-ECO-R1 from Escherichia coli, creating a site for the insertion of the targeted DNA sequence. Subsequently, DNA legacy seals the DNA fragments together, generating the recombinant DNA molecule. Now, how does recombinant DNA relate to inheritance? One important aspect is sex linkage. In many organisms, including humans, sex is determined by specific chromosomes, which are the X and Y chromosomes. Females typically have 2 X chromosomes, double X, while males have 1 X and 1 Y, XY. Genes located on these sex chromosomes are called sex-linked genes. Since the Y chromosome is much smaller and carries fewer genes, most sex-linked traits are associated with the X chromosome. To help you visualize sex linkage, consider an X-linked recessive trait, like red-green color blindness, because males only have 1 X chromosome. If they inherit the colorblindness allele, they will express the trait. Meanwhile, females need to inherit the colorblindness allele on both X chromosomes to be affected. This difference in inheritance patterns explains why X-linked recessive traits are more common in males. But where does the variation in these traits come from? This brings us to genetic recombination. During meiosis, the cell division that produces sperm and egg cells, homologous chromosomes pair up and exchange genetic material. This process, called crossing over, is a form of natural recombination. It shuffles the genes on the chromosomes, creating new combinations of alleles. So how does recombination affect sex-linked genes? Imagine a female with 1 X chromosome carrying the colorblindness allele and the other carrying the normal vision allele. During meiosis, crossing over can occur between these X chromosomes, resulting in egg cells with different combinations of these alleles. This means that the offspring can inherit new combinations of sex-linked traits. Now, how do we study these genes, including those involved in sex-linked traits? This is where recombinant DNA technology comes in. Through isolation and manipulation of specific DNA sequences, scientists can map the location of genes on chromosomes. This process, called gene mapping, helps us understand the organization and function of the genome, including the genes responsible for sex-linked traits and other genetic characteristics. Is recombinant DNA restricted to gene mapping? Of course not. For instance, in medicine, recombinant DNA technology is used to produce therapeutic proteins such as insulin and growth hormone. It can also be used to develop gene therapies. Agriculture benefits even more, as recombinant DNA technology has paved the way for the creation of genetically modified crops with enhanced traits such as increased yield, pest resistance, and improved nutritional value. So, let's bring it all together. We've seen how recombinant DNA techniques allow us to work directly with the genetic material itself. It is important for understanding how traits, including those linked to sex chromosomes, are passed down from one generation to the next. We also explored how recombination during meiosis mixes up genes, creating new combinations of traits and contributing to the diversity we see in living organisms. Not only that, but we were also introduced to the applications of recombinant DNA technology and how we benefit from it. But before we end this discussion, I want you to think about how your genes, like a story written through generations, is constantly reshuffled by recombination. How does it change how you view your place in that ongoing narrative of inheritance? I'll leave you to ponder on it and hope to see you on our next topic.